All right, good morning everybody. How are we all doing? Good? Great. All right, so we're going to continue the lecture series, and the next lecture is going to be on again a more continuation of the basics of electrochemical biosensing devices. Yesterday I did mention to you at the heart of the biosensing device is a bioreceptor. So today we're going to look a little bit more on the bioreceptors. We're going to do a lot of recap today just to kind of consolidate on what we learned yesterday. And I'm going to focus a lot today on one of the bioreceptors, which I think is going to be a future for tomorrow. Right? It's being used quite a lot, and it's got a lot of potential. And I'm hoping, and I think my prediction is that this is a bioreceptor which will be a future for tomorrow. So if I showed you the course structure, we are on day 2 where we're doing electrochemistry fundamentals and understanding the electrode miniaturization process, focusing mainly on the bioreceptors and how the bioreceptors can be tapped into to exploit the sensitivity. And then there's also going to be a lecture after this that will be mainly on epidermal. So I've moved the lectures around because tomorrow I want you really to be very fresh to give you one of the best lectures that I think is going to be here, which is going to be combining bioreceptors together to get more extraordinary sensitivity, getting that ultra sensitive detection that you normally expect from your biosensing devices. I do appreciate I have a wide audience here. Some of you are not working directly on biosensors, but as I mentioned yesterday, biosensing technologies are enabling technologies. You can extend these biosensors into your area of application, whether you're working in the area of forensic sciences, environmental sciences, healthcare sciences, whatever it is, you can definitely extend the applications there as well. So the lecture is, as I said, this is the learning objectives. We're going to be understanding electrochemical biosensors again. We're going to be looking at the different types of sensors, how they are fabricated, the transduction principles once again. We already covered a little bit yesterday. We're going to consolidate on that. And then we're going to look at some real world applications here as well. Suggested reading. This is one of the favorite books that I have, which is on analytical electrochemistry by Joe Wang. Fantastic book. There's also a paper which was one of my first papers on the use of subdermal devices, wherein we actually got the subdermal devices investigated on a microfluidic device. Microfluidics, as you all know, is where you have miniaturized channels which are used for manipulation of the flow of liquids. You can use that for having multiple reservoirs, mixing them together. So this is a nice paper that was published, I think, back in 2012. So it's a good reading as well. So I would suggest reading these 2 papers, and they will be made available through the organizers in a zip file where you have all the collection of all the papers. So just a quick recap. Yesterday we saw what are chemical sensors and how they become biosensors. We saw that a chemical sensor is mainly a device that would transform chemical information. Mainly we're looking at concentration. That's what we're mainly interested. In some cases we also look at the activity as well. So for example, when we look at kinases, CDKs, we are looking at not just the concentration of, but we're looking at the activity, and we measure the activity as well. But the most general example that's given is that a chemical sensor is a device that would transform chemical information, mainly concentration, of a specific sample component into an analytically useful signal, which could be in the form of a visual signal which you see as a reading on your device, or which could be an alarm. For example, if you have an oxygen depletion sensor here, which is giving you a warning that the oxygen levels are dropping down. And then, as I mentioned yesterday, your chemical sensors would have 2 basic components, and these components are normally connected in a series. You would have what we call as a molecular recognition element or a receptor, which is combined to a physicochemical transducer, and the physicochemical transducer would then convert it into a useful analytical signal there. Now this receptor becomes a bioreceptor, and when this becomes a bioreceptor, it becomes a biosensor. Right? A biosensor is a sensor which would normally have a recognition element which involves or which utilizes biochemical mechanisms. That's the example of a biosensor. So we saw examples. I gave you, we went into depth. I think we're going to be covering that again, but we saw examples of how enzymes could be used for making biosensors, how antibodies could be used for making biosensors. We are basically exploiting the specificity of these bioreceptors to get your biosensors of interest. What exactly is a biosensor? As I said, again, it's an analytical device which has got a biological recognition and signal transduction element, and it measures a signal proportional to the analyte. That's the important thing here, that your biosensing device should give you interesting information or useful information which is on the concentration, and this concentration has to be directly proportional, or the current values that you measure should be directly proportional to the analyte concentration that you're measuring here. That's what makes it very important. And the common targets could be, you've seen examples of glucose. You could have stress hormones such as cortisol, or you could have cytokines, as I mentioned, CDKs, examples of cytokines as well. IL-6 we saw yesterday was another example of cytokines. I showed you this slide yesterday as well. I said how we have different samples here. These are your different samples on your left hand side, and then how you could use the transducers, and your transducers which are involved in the signal transduction would have your bioreceptors, which could be nucleic acids, cells, antibodies, enzymes. These are your bioreceptors, and then you can have devices, or you can have electrical interfaces such as your field effect transistor devices that we saw. Somebody asked a question about yesterday. Nanowires, I said to you that these are very fine structures which have got a very high surface to volume ratio, and therefore very useful and give you very good sensitive detection there. You could also have nanoparticles. Gold nanoparticles and silver nanoparticles have been used. You might have heard about this technique called as surface enhanced Raman spectroscopy, right? That's where you use cracked gold, cracked silver, which gives you ultra sensitive detection there, but that's an optical detection method. That's why we're not going to be discussing too much on that, because we are focusing mainly on electrochemical biosensing methods. And you can also have electrodes. This is normally people who work in the lab who do cyclic voltammetry and things like that have already got used to this particular setup there. So this is your signal transduction. This is your signal transduction is done. Once this is done, then it's transferred to something that's what we call as an external circuit, which normally has a signal amplifier to amplify the signal to get a measurable current there, and a signal processing which can make sense, do the conversions, and then finally you have a display system which is part of your electronic system. So these are the main the components of an electrochemical biosensors, and these here are the different types of samples. I won't go much into details. We've covered that in much detail yesterday. Again, this is a recap of what we just saw. You normally as I said, the heart of your biosensor is your bioreceptor there. The bioreceptor is what gives it the required specificity. It could be an enzyme, an antibody, an aptamer, and I've introduced this new term here, MIPS. We saw a little bit of this. This is basically molecularly imprinted polymer. Right? We'll see examples of this in greater detail today when we'll have a look at MIPS, and I'm going to be spending a lot of time today discussing about MIPS, because as I said, they have potential to be the future bioreceptors, because they allow you to, and they also call as plastic antibodies. All right, we've already seen this. We move to the next slide here. This is a different slide here. The bioreceptor types that we've basically covered, or which we have understood as part of this GAN series, are enzymatic sensors, enzymatic bioreceptors, immunosensors will have antibodies as bioreceptors, aptasensors will have aptamers or strands of oligonucleotides, and MIPS will have molecular imprinted polymers there. Right? These are the different types of bioreceptors that you will normally come across, and you can see here this is your enzymatic bioreceptor. It works according to the lock and key model hypothesis that we have. This is what gives it the required specificity that we need for monitoring or for biosensing. This was a slide that we covered in greater detail yesterday. Everybody happy with it? Anybody wants a recap of this again? So we saw yesterday how we have different generations of glucose biosensors. We have the first generation where we are generating hydrogen peroxide, right? You don't have any mediators there. This is the first generation, but then to break this hydrogen peroxide and release electrons to be able to measure current, we are working at very high potentials, and therefore to reduce those potentials, we normally work with a mediator. Mediators work at lower potentials, and that helps you in addressing the problem of interference, right? And there's also the third generation there, where you have a direct electron transfer from the cofactor, from the enzyme cofactor to the metal surface there as well, right? So this is something that we covered yesterday, and we also discussed a little bit about the pioneering works that were done by Leland C. Clark towards the first generation, Professor Tony Cass towards the second generation, and Professor Koji Sore done towards the third generation of electrochemical or enzymatic biosensors there. Similar to the glucose biosensors, you can also have lactate sensors, which could be first generation, second generation, third generation. In fact, in the case of lactate biosensors, anybody working on lactate biosensors here? You have the third generation which is available. You have a direct electron transfer sold by a company called Lactazyme. Right? If you want to work on that, you can use that enzyme, and there are papers that are coming out from Imperial College where they've used the third generation lactate biosensors, which are commercially available there. If they're commercially available, they're clearly deemed to be safe. All right, This is a slide which is showing you about the electrochemical immunosensors. As I said, when you see the word immuno, that means you're talking about antibodies here, right? And how do you represent antibodies? They are normally represented in the form of a Y shape, right? These antibodies, as you might have already studied during your school time biology and chemistry lessons, these have got very good specificity, and the interaction between the antigen and antibody is very, very specific, right? These are normally, we've been normally this main source of your antibodies that are normally natural antibodies is animal, right? So you normally extract them out of animal. It's a very, getting the antibodies out and getting, doing the purification of these antibodies. It's a very, very expensive process. It's a very tedious process. It's got multiple steps there, and it really makes the whole process very expensive. Enzymes as well. I mean, even the enzymes, when we buy the enzymes, they are really expensive. They're not that cheap, right? Antibodies, again, they are more expensive than the enzymes. It depends on the antibody you're looking for, but the rare antibodies or rare diseases are much more expensive because it's very hard to extract them and purify them and to get them to a form which can then be used for immunosensing applications. So what you see on your slide here is an electrode material which should be a metal surface. On the metal surface you have a nanomaterial modification. This is something which we're going to cover tomorrow, wherein we're going to talk about combining nanomaterials and then having the enzymes or having the bioreceptors immobilized on them. What is the advantage of them? We're going to cover a lot of this tomorrow because I want to keep this very simple right now. I just want to focus on single bioreceptors in this lecture, but tomorrow we are going to be looking at something really interesting, how you can combine the bioreceptors, with nanomaterial systems, and how you can combine the bioreceptors amongst themselves to get some very interesting results there. Okay, so this is basically what we're looking here is you have your different nanomaterial modifications. This looks like a dendrimer, and this looks more like a graphene kind of thing, and this looks more like a carbon nanotube here. Right. So what you're doing here is you have your antibody which is immobilized on the surface, right? And once you've immobilized that, this particular technique is called as ELISA. Anybody heard about ELISA? It's enzyme-linked immunosorbent assay, right? So what is happening here is you basically have one antibody, and then you have a target molecule, and then you have a second that comes in. So your target basically is sandwiched between two antibodies, and then this secondary antibody has basically got a fluorophore there, or an electrochemical tag there, and if it's got a fluorophore, then you can measure the changes in fluorescence, and if it's got a electrochemical tag, then you can measure the changes in the electrochemical properties that you have here. So these are typical examples of how an electrochemical immunosensor would normally work. The link is down there, and you'll be getting the slides so you can follow it up. The next example I'm going to talk about is APTA sensors, right? Aptamers, basically strands of DNA, strands of oligonucleotides. Right, and we all know in nature, we know that we have the double helical structure, and this double helical structure, the two strands are held together by hydrogen bonds, right? You have the adenosine pairing with thymine and cytosine pairing with guanine, right? Cytosine pairs with guanine using how many hydrogen bonds there? 3. Well done. And adenosine pairs with thymine pairing by 2, right? So you have 2 hydrogen bonds there. Well done for remembering your school level, whatever we call it, GCSE level biology and chemistry there. Well done. So that's basically that's basically how your oligonucleotides normally bind amongst themselves. But at the same time, they're also known to bind to certain molecules, and it's based on the same principle. They use their hydrogen bond capacity, and they're able to bind with certain molecules of interest, and they are very specific as well. They give you the desired specificity. As I said, bioreceptor, heart of the biosensor, has to give you the desired specificity, and not only give you the desired specificity, also give you the desired sensitivity as well, right? These are terms that you can, specificity has to be natural, but selectivity, the sensitivity is something that we can fine tune as well, depending on what we are trying to measure and what concentrations or what's the dynamic range of the biomarker or analyte of interest is in. So this is an example of how your aptamers can be assembled on gold. Anybody heard about self-assembled monolayers? SAMs? So thiol groups, you know, alcohol groups are OH, thiol groups are SH groups, right? So if you have a molecule which is terminated with a thiol group, so if you have a molecule and then you terminate with a thiol group, the thiol groups are known to form very nice layers of self-assembled molecules, right? You can have them literally because of the, you know, the forces balance out each other. The forces of attraction and forces of repulsion, they balance out each other so nicely that they stand they self-assemble themselves in a very nice uniform layer, right? So these are your thiol compounds, right? Once you know how your thiol compounds are, you can have your oligonucleotides which have been terminated with a thiol group. So as soon as you drop your thiol groups, your solution of your thiol onto a gold surface, they will all stand on it and assemble themselves, keeping the equal distance spacing there. You don't have to do anything. This is all done naturally. You don't have to assemble the molecules one by one. It's just all done naturally. And there are some beautiful AFM pictures that you can pull out from the internet, where you can see that these molecules are standing really nicely next to each other, right? Very well assembled, very good spacing, very uniform molecules. So this is something that you can exploit, and you can use that. So what you see here is basically the S is basically your sulfur groups from the thiol, and you can see that it's standing down as self-assembled monolayers, nicely assembling on the gold surfaces there. And then this is what you have here is basically your target molecules, and you can see that what happens is when it's standing straight, as soon as it binds with something, it gets a little bit heavy, and then they start to bend downwards. So as you get the electrochemical tag closer to the surface, you're basically facilitating a much better electron transport. To have electron transport, you remember when we were talking about third generation glucose direct electron transfer, we were talking about how the electron transfer groups have to be closer to the surface, to the metal surface, so that you can have an efficient electron transport there. I mean, what is electrical conductivity? It's the flow of electrons. How do you ensure that you have a good flow of electrons? You have to ensure that your electrochemical tags are closer to the surface, so that you can have a much efficient. And there's a certain value there. If you look into papers that talk about direct electron transfer, you will see that they come up with values which are normally in the range of very few nanometers, which allow where the electron transfer takes place. If you look at glucose oxidase, if you look at the molecule of glucose oxidase, the cofactors are all quite high, sitting quite high up there. So what they do is they normally fragment it and bring it closer to the surface. So that's why your direct electron transfer enzymes are normally engineered enzymes. They are fragments of the enzymes, because you try to bring the electroactive part closer to the surface, so that you have a very efficient electron transport there. All right. So that's your. Did I just jump? That's your electrochemical aptasensors. With that, now I take you to what I would say is a little bit of a case study kind of a thing on a new class of bioreceptors, which we call as molecular imprinted polymers. I'm not very enthusiastic about it just because I'm working on it personally I'm working on it myself. It's also because of the fact that these are actually have been reported in the literature as well to be very sensitive as well. They can, and the reason I love them is, number one, my objective is to make low cost devices, right? If I can make something which is low cost, something which is stable, if it's something that can be made by scalable technologies, what else do I need to make low cost devices? As I said, antibodies, problem is you're deriving them from animal sources. Extraction and then purification, making it really expensive. Enzymes, you're deriving it from plants. Again, extraction and purification. This is something that can be synthesized, and you can make them as you're building your biosensors. You don't need to buy them separately. You can buy the monomer solution, and then you can polymerize it. And there are, polymerization is those of you who studied chemistry know polymerization has been going on since ages. We know about all these wonderful natural polymers that are existing in the nature, and we also know about the harmful polymers that exist that we have created. Your polythene bags and all that stuff. These are polymers that are you can choose to be in accordance to green chemistry and biocompatible as well. All right. So molecular imprinted polymers are also referred to as synthetic antibodies, because you are synthesizing them yourself in the lab, or they also called as plastic antibodies. You can modify their properties as well, right? The main advantage is you can tailor the specificity, right? If you can take a monomer, you can mix monomers together, and you can get the desired specificity. You have a monomer which is giving you a particular shape, but it's not giving the required specificity. So you can have another monomer and another polymer around it to alter the properties, to give it the desired shape, to maintain the shape. It's very important here, because you're creating cavities, you're creating binding sites here. It's very important that you maintain that the whole binding site in its native form, right, the way you create it. Right. So the advantages of molecular imprinted polymers is, number one, it gives you tailored specificity, right? You can tailor the specificity to go down to lower concentrations as well, and I'll give you examples in the next few slides as to how we have used these molecular imprinted polymers for early diagnosis of a disease called Alzheimer's disease. Anybody knows about Alzheimer's disease here? Alzheimer's disease is a neurodegenerative disorder, right? It happens, I think 70% or 80% of dementia that happens with old age is because of neurodegeneration of the nerves, right? It's a natural process, right? But the problem is that it's becoming more and more common. Again, as I said yesterday, we are all victims of our own success, right? As we have managed to bring in technologies that have increased our longevity, and we are all living longer now. Earlier people used to die by the time they were 65. I'm talking about 1930s, 1940s, 100 years back, right? Now we all know that the average average age is around 80, and dementia starts around 75, 74, 75. So we are all worried about the quality of life, because there's a lot of dependency in that particular thing. When people do not have good control of their nervous system, they need help there, right? That's where, so we need to be able to diagnose early, and now we have medication for management of Alzheimer's. We need early interventions. We all have the medication, but we need to know when do you start the medication. So for that, you need to be able to diagnose the disease. And actually there are reports which have clearly suggested that if you have the right diagnostic tools, you can actually diagnose Alzheimer's disease 15 years in advance before the onset. You can look at the pattern of the neuronal degeneration that's taking place, and you can predict when it's going to start. And then if you start the therapeutic intervention at the right time, you can then ensure that it's managed in a nice way. You have a good quality of life. Right? There are ways, there are different types of studies and tests there. So coming back to these molecular imprinted polymers, so what I was trying to highlight here is that we have used these molecular imprinted polymers for our project on Alzheimer's disease to basically develop early diagnostic systems. And as I said, you can tailor the specificity, and they're highly stable. So we have a UK-Japan project. So we made our devices with molecular imprinted polymers, and we sent it by courier to Japan. And when we tested them there, they were all working very fine. So they're all very stable, right? If you look at the objective should always be, as they say, the world is a family. So you should always try to think of developing devices that not only work in your own conditions, but also which can work in global conditions. You should think about continents like Africa, where you have extreme heat, the sub-Saharan heat temperatures or heat conditions. You have high heat, high humidity. Even in India, you go from region to region, the temperature changes, the humidity changes. So you should have a device that not only works in Kashmir, also works in Kanyakumari, where you have the whole extreme that you have. Not only works in Gujarat, also works in Assam. You should be able to capture. Your device should have that robustness, should have that stability, that it works in all conditions. And that's one of the things that you get with molecular imprinted polymers. That's why it happens to be one of my favorite bioreceptor. So it's highly stable, it works in robust and harsh conditions, and as I said, it's very, very cost effective. You can start making them in the lab. When you're building your biosensor, it's not about, you don't have to order it as a separate chemical. You can buy the monomer and you can polymerize it yourself. And the monomers are not that expensive. Nothing compared to the prices that you pay for enzymes. I don't know what the prices of enzymes here are for glucose oxidase, but we pay a lot of money for glucose oxidase, lactate oxidase, and if it's hydroxybutyrate, it's much more expensive. So they're very expensive. Antibodies, we don't even bother looking into antibodies because we know they're very expensive. ELISA kits are based on antibodies, and we know that it costs a lot of money. For a plate for Simoa analysis, which is single molecule analysis using, which is basically a glorified form of ELISA, that's about 6,000 pounds. You can do the maths now, multiplied by 100, how much it costs to do 48 samples, right? So it's very expensive. This molecular imprinted polymer gives you a much more cost-effective solution here. Right? You can use this, it's much more cost effective, and as I said, durable, and they give you a long shelf life. You can store them over a longer period of time. People who work on enzymatic sensors, where do you store your sensors? 4 degrees in the fridge. Can you imagine transporting the sensors if you were to do an experiment somewhere in Kampala or in Accra? Can you imagine how your sensors are going to work? Right? So transportation is a problem. I think that's the problem we had with vaccines, the cold chain thing, right? Anything that needs to go into cold chain, that's a problem there, because then you need extra resources to be able to maintain them. You're doing all this huge program there, and then you don't know whether your vaccine is still efficacious, whether your sensors are still working or not. So you need something which is more stable and which can withstand all the harsh conditions. So this one is, as I said, is durable and has a long shelf life. You don't have to store it in your in the fridge. The cartoon that you see here, there's a simulation here which is done. This is fantastic simulation, has been done by my fantastic PhD student Sudhansh Deshpande, wherein he's basically showing you, what he's showing you here is this molecule in the blue. This one is your vancomycin. It's showing how vancomycin comes and binds to this molecular imprinted polymer, which is basically a plastic antibody designed for the vancomycin molecule. This is your cavity. You can see here, it comes and nicely binds there. How this all happens, I'm going to be showing you this in the next few slides. So, how do we fabricate molecular imprinted polymers? I'm sure most of you must be keen to know that, since I've told you so many nice things about it. What you do is you start with a template molecule that you see on your screen here. This could be your target biomarker, right? This could be a drug for which you're doing therapeutic drug monitoring. This could be a large molecule. This could be a biomarker of Alzheimer's disease. This could be a biomarker of brain infection. This could be something as simple as insulin. This could be something as small as glucose molecule, right? So you have your template. So you start always with the template, because you're creating, as I said, molecular imprinted polymers. You're creating a template here. How do you create a template? You take your target molecule, your molecule of interest, right, which is basically your biomarker, and then you bring it in, you put it in a solution of your monomer here, right? What happens here? You will see that, I will show that in the next slides. Your monomers will interact with these particular template molecule according to the size and according to the charge. So in the first step, that's what happens, and that's a very quick thing that happens. And then what you start doing is you cross-link your monomer so that they then start to polymerize. Cross-linking means that you're binding your monomers together to form a much more rigid structure around your template molecule, which is your biomarker. So in this third step, you have cross-linking of your monomer to form the rigid structure, and then what you do is you eat away this template structure that you have here, and then what you end up getting is you remove the template and you get your cavities, which correspond not only according to the shape but also to the charge centers. Every molecule, if you look at large molecules, every molecules have got charge centers, right? What are these charge centers and what are the interactions I'm talking about? We'll be seeing that in the next slides. But you need to understand that you need to conserve the size, the shape, the geometry, the charge centers, all of these things together to be able to get a template into which you have your biomarker comes and fits in, right? You may have a complex mixture where you may have molecules with similar size as your template, as your biomarker. They will compete against each other. Therefore, you need to make your template very specific, and that's can only be done by not only conserving the shape but also conserving the charge centers there as well. Okay, so what are the proposed interactions when you have your interaction? So I mentioned about this step here where you have interaction with functional monomers here. What helps in stabilizing it? You have, so if you talk about a polymer, a monomer such as phenol red, which is one of the monomers that we are using in our group, and we've also managed to file some invention disclosures on these. So we have basically got this phenol red. Basically, you have the hydroxy groups. You also have the sulfonyl oxygens, which are strong hydrogen bond acceptors. So you have these the hydrogen bond formation which takes place between phenol red because of the hydroxy group and the sulfonyl oxygens that you normally have. Hydrogen bonding, everybody knows about hydrogen bonding here, where every time you have hydrogen placed between a electronegative atom such as fluorine, nitrogen, oxygen, you will find hydrogen bonding there, right? So here, phenol red basically has got these two groups here, hydroxyl groups and the sulfonyl groups, which are so at the hydroxyl group, clearly you have the hydrogen between the oxygen and there's going to be a next electronegative atom there. Sulfonyl groups, the oxygen atoms are going to be acting as strong hydrogen bond acceptors there, right? So that's what happens in the case of sulfonyl groups here. In the case of, so this is what hydrogen bonds, this is what comes from the monomer. And if you look at the target analyte, which in this case is vancomycin, you have multiple hydroxyl groups, you have the amine groups, you have the amide linkage. I'm doing a little bit of chemistry here just to kind of explain things, what is happening at the molecular level, right? You have the amide linkage, you have the carbonyl groups in the carboxylic acid group, and then you have the ether linkage. Yeah, just delving down deep into what's happening here, what are the proposed interactions here. So you have the interactions of the hydrogen bonds, which, is it a good thing or a bad thing? It's a good thing. It helps in stabilizing because hydrogen bond is a form of an attractive force, right? It's binding it together, right? If you have forces, you may also have other forces which will be responsible for repelling it, for destroying the structures. You will also have that. You have to find the right balance there, right? So these are the hydrogen bonds that take place. You will also have hydrophobic interactions. You will have phenol reds contribution. You have got the phenol groups, right? The more carbon content you have, the more is the hydrophobic interaction there. So there's going to be few hydrophobic interactions from the phenol reds contribution. And in case of vancomycin, you again have these alkyl groups and the aromatic rings, which will also contribute towards the hydrophobic interactions, right? So the presence of hydrophobic pockets can then lead to phenol red being lightly bound to the sequestered hydrophobic packets, right? That's the kind of interaction, and this interaction is driven by the tendency to exclude water molecules. That's an important thing that we need to look into when it comes to hydrophobic interactions. At the same time, there are also going to be another form of interactions, which are your electrostatic interactions. As the name suggests, starting with both because of the charge and the differences in pH. All right, both molecules will exhibit different properties at different pH, so you need to make sure. So you normally, when you're doing this templating thing and you're trying to create the template, you want to see what are the different parameters you're trying. What is the optimization parameters here? You'll try different pH, you will try different ionic strengths, you will try different concentrations, different ratios of your monomer to your target molecule, and see what is giving you the best combination. This is what is normally developed as part of your biosensor development thing. This is what normally people who work on biosensors, this is what they optimize to get a device which gives you the desired sensitivity. This is where I say sensitivity is something that you can play around with. You can try to make your devices more and more sensitive by making more and more sites available for binding, making more and more efficient sites, and making it more and more specific. Right? This is what basically what we normally work towards optimization. So then once you've done all of that, then you move to the sensor usage. This image that you see here is basically a PCB electrode here. What we need is just 70 microliters of the drop. You wait for 30 seconds, you take a measurement. I think it's 30 minutes. So you're allowing all the binding to take place, and then you take a measurement, right? So you get a cyclic voltammogram there. So basically what you need is, there's no sample preparation required, there's no washing step required, there's no external redox probe required here to probe the electrochemical properties here. That's the advantage of using this particular polymeric system. And look here, this is the dose response curve. So what we've seen, this is what's been optimized. So I'm showing you the best result here. I'm not showing you all that hard work that's been done in the back office, which is mainly done by Sudhansh himself. I'm just showing you what the final dose response curve looks like, and I'll explain you why I like this dose response curve. Here, your MIP material here happens to be polyphenol red, right? So you can have phenol red, which is your monomeric solution. You polymerize it to polyphenol red by cross-linking it. You're getting a nice isotherm here. You get the sample matrix here is, we work directly with spiked serum samples. Where do you get your serum from? You get it from your blood samples, right? So the serum samples have been obtained from blood samples, and we've spiked them with different concentration, and you can see that the linearity is all the way up till here. This happens to be the linearity range, right, almost till here. This linearity range is about 70 micrograms per milliliter. And actually what we are looking for, this is for your p-tau proteins. If you look at the lower limit of quantification, it's 2.7 micrograms per milliliter. Upper limit of quantification is 70 micrograms per milliliter, right? How is this useful? Now we know that the typical value, the typical mean value or the median value at which you would get a full diagnosis of p-tau proteins is around 4.8 to 6 micrograms per milliliter. Normally it's 4.8 is the cutoff, but what happens is you can have people with no AD, no Alzheimer's disease, but can have a high concentrations, because this is related to your age as well. And that's why at some point I will explain how we need to bring in the machine learning and the AI models as well, right? Because you have this whole thing, the baseline, right? So all of us have some form of neuronal degeneration. At the moment our values are sub micrograms per milliliter concentration here. So this is basically showing you the limit of detection here. So this is the concentration, and we are getting the desired sensitivity here. All right. So this is a performance. Sorry, my apologies. I'll take my words back. This is about vancomycin. Right. I confused you. I'm sorry for apologies for confusing you. This is about vancomycin. So vancomycin, here you are looking at micrograms per milliliter concentration. sorry, this was picograms actually. This was for Alzheimer's. So my apologies. I'll just take this back. This is for vancomycin, and that's why we are looking at micrograms per milliliter concentration. This is the typical example which I gave you yesterday of therapeutic drug monitoring. Do people remember that? Therapeutic drug monitoring I gave you? I told you. So what happens is drugs, normally if you look at the concentration, you normally would have concentration over time. For a drug to be effective, you normally administer a drug, it goes into your body and it distributes itself to reach a peak concentration and then it drops down. So before this drops down, you administer another drug, so it goes up and then drops down, and then you administer another one here, it goes up and drops down. So what you try to do is you try to maintain this concentration level here, right? And this range at which it's normally effective is called as the therapeutic index, right? You have to get your drug concentration in the blood in that particular range for it to be effective, right? And we saw the example of antimicrobial resistance. Overuse and underuse can cause antimicrobial resistance. So it's very important that you stay in this therapeutic range, right? The therapeutic range for vancomycin happens to be between 1 to 10 micrograms per milliliter, right? And what we can see here is you have the lower limit of quantification is 2.7 micrograms, and upper limit of quantification, it happens to be 70 micrograms per milliliter. That's how these normally work, right? So it's a good example wherein you got the desired range here, just with phenol red polymer here. So that's an example of vancomycin here. I'll just make it very clear. This is vancomycin therapeutic drug monitoring in serum samples, because when I saw serum, that's when I understood that I was doing a mistake here, because you normally measure vancomycin in serum samples, not in plasma samples. In the case of Alzheimer's disease, you measure the biomarkers in plasma samples, and in this one you measure it in serum samples. So what are the main challenges in translational research when you have bioreceptors such as your molecular imprinter? Cost of screen printed electrodes is very high. This is a PCB electrode, right? And these ones you can get them for a price of 0.09 pounds or 9 pence, right? Screen printed electrodes, how many of you have worked with screen printed electrodes? When you go and buy them, you get a box, and that box is about what, 400 pounds? 400, 500 pounds? So you're talking each one of them costing about 5 to 10 pounds for the screen printed electrodes. And screen printed technologies are normally considered to be scalable technologies, right? We didn't find them very scalable, we found them to be more expensive, and as I said, the objective of my research group is to develop low cost diagnostics for global healthcare solutions. I can't do low cost diagnostics if I'm getting something as expensive as 5 to 10 pounds. I would prefer something which is costing me 9 pence. Even if it's made in some remote corner of the world, I would prefer to source them and I would prefer to buy them. Yeah, or probably make them in my own lab if possible. Okay, so the cost of screen printed electrodes is very high, therefore we introduce these PCB electrodes that you see on your screen. As you can see here, if you look at this one, you basically have got a reference electrode, you have a counter electrode. This is twice the size of the working electrode, and you have 2 working electrodes here, so that you can do multi-analyte detection. You can also do something called as redundancy. This is the term wherein you measure the same analyte but on 2 different electrodes, so that if one of them goes wrong, at least you've got a value there, and you can also take the average values there as well, right? Sometimes it's very useful there to look at the reproducibility of the device as well. I'll show you devices where I think the devices that we are going to be working, they've probably got 4 electrodes, right? So you can also have 4 electrodes. You can use them for the same analyte to get n equals 4, or you can use them for 4 different analytes. The choice is yours. N equals 4 happens to be a very good set of data there. People who work in analytical chemistry, any analytical chemists in the audience? No? You understand the value of meaning of n equals 3? Rishita, n equals 3. If you have present any data which just doesn't have n equals 3, you don't have the paper accepted. You need to have n equals 3 and you need to show the error bars, the variation between the sensors as well, right? You have to show reliable, reproducible sensors. Right? So having 2 of these working electrodes helps in getting that. If you have n equals 4, even better. Minimum you need is n equals 3, because if you have n equals 2, you can have 2 data points, 2 points, and you can always get a straight line between those 2 points. The other things that problems we have is you also have biofouling effects on the surface. This is where we've used highly porous gold, electrochemical interference from other compounds, and expertise you need for data analysis. This is just to show you how the data analysis is done. These are the traces you normally get from the two different working electrodes, and then what you can see here is this is showing you how the data is collected. This is for vancomycin, PPHR. You can see this is patient 1, just to show you how we are gearing up for the human studies, right? So the results I showed you are studies that have been done in plasma, but in vitro conditions. These are not real samples. We've taken plasma, so that's how you normally get your dose response curve in the lab, right? You get the blood is sold as a reagent in the UK. I'm sure it's sold as a reagent in India as well. So you can buy blood which has been tested for different pathogens. You can use that blood. You can spike it with different concentrations, so that then you know this concentration gives me this value, and then you can get a dose response curve, right? So this is an example where then he's getting the software ready on data analysis, and basically this is an example of a slide where we did clinical samples for n equals 12, and this is the range that we get here. As you can see here, we had patients who were given vancomycin, which is the last resort antimicrobial, but they were also given a cocktail of drugs, because before giving them vancomycin, they were given different anti-TB drugs: meropenem, piperacillin, tazobactam, isoniazid. All of this was given to them. So there would be some traces of that as well. So you would expect them to be electrochemically active and some interference from these as well. But we managed to use the right set of conditions using differential pulse voltammetry and identify the exact potential we should be working on to get to identify the peaks and find out which is the best working condition here, right? Now this result is basically, this is the value that has been predicted by the molecular imprinted polymer, and this is the value that has been predicted by LC-MS. LC-MS is liquid chromatography with combined to a mass spectrometry, right? This is normally used for, it's a standard method. It's a lab technique. It's something, if anybody who's seen a LC-MS or a GC-MS, you will see these are quite big. I mean, they're benchtop, don't get me wrong, but these are not portable devices. So what we are trying to develop are portable devices which we can carry around. We can work in any part of the world, and you can work on farm side, you can work on bedside, you can, point of care kind of devices. That's the kind of things. So these are clinical samples against which we validated our devices, and you can see beautiful correlation there, and there's a good agreement between our devices, our biosensing devices, and the LC-MS data that we've obtained here. So what's the elephant in the room? Well, MIP specificity. I mean, do we need our MIPs to be highly specific, highly selective? We need it, right? I mean, the data I showed you was showing more on the lines of micrograms per milliliter concentrations, right? That's fine for vancomycin, that's fine for this application, but there might be applications where you have to go down quite a lot. For example, Alzheimer's. When you're doing early diagnosis, you're trying to predict something 15 years before. You can imagine if you did a measurement in my blood, still far away from my 70s, there would still be some traces. It's going to be trace analysis, but we would like to have sensors which can look at my concentration and then track it and extrapolate it and tell me when do you think Alzheimer's will set in me, and then I can start my medication accordingly earlier on and things like that, right? So the specificity is still in infancy. That's what we think. They may have low specificity when it comes to molecules with similar structures. That's the problem here, right? So you've got to be very careful with the choice of molecular imprinted polymers, and there are a lot of materials which can be easily polymerized to give you the templating there, okay? And then the molecules, they may not be suitable for all drugs, because some of the drugs are highly hydrophobic. They're not soluble in water, and what I showed you right now was mainly how you are electropolymerizing them. And if you're looking into doing electrochemistry on them, it has to be aqueous based. You can't have organic solutions there, right? So some of the drugs could be hydrophobic drugs. If you have hydrophobic drugs, they may not be soluble in water, right? That's a problem there. It's not a one for all kind of a solution there, although we've tried to see how many we can do, but you will have to develop your molecular imprinted polymer template according to your biomarker, according to your template molecule, according to your biomarker molecule. It's not like you take polyphenol red, it works for everything. You will have to fine tune its properties. You will have to change its specificity, right? And then there's also this, we are undergoing some continued research on further developments here, and it's also suggested that we combine the receptors, and we also use the dual receptor approach there as well. Sometimes that helps in enhancing the sensitivity, and this is something, please come tomorrow morning as fresh as possible, so make sure you have a good night's sleep, no partying today. We're going to have a real good lecture tomorrow on combining bioreceptors to get ultra sensitive biosensors. Yeah. Okay. So looking at the conclusions based on the molecular imprinted polymer work that was done, vancomycin is a glycopeptide. It's an antibiotic which is a last resort antibiotic. Therapeutic drug monitoring is essential for vancomycin because the therapeutic range that we have is 15 to 25 micrograms per milliliter. It's high and it varies from individual to individual. This can go down to 10, as I mentioned to you before. This can go down. So if you have something where you're trying to measure between 15 and 25 micrograms, you would want to have a dynamic range of 0 to 70 micrograms per milliliter concentrations. That's how you normally design your biosensors. So if your biosensor is giving you the range of 0 to 70 micrograms per milliliter, then you know it's something that's going to be working very well. All right. MIPS can be made out of polyphenol red. This is a new material which has not been used before for making MIPS. So it's the first time we employed these polyphenol, or we employed phenol red monomers to make molecular imprinted polymers. And then what we have also seen is you have different types of interaction between the vancomycin and the molecular imprinted polymers. There were the hydrogen bonds, there are the hydrophobic interactions, and there is also the electrostatic interactions. The dynamic range we saw is from 0.848 to 70 micrograms per milliliter, which is very good, which is giving us the desired sensitivity. We are covering the desired dynamic range, which is fantastic, and we have validated these samples against something which is the gold standard, which is liquid chromatography and mass spectrometry system there. All right, that was all about molecular imprinted polymers. I hope I've given you a good flavor, and I hope I've convinced you why, if you were to choose one bioreceptor to start off working with, it would be molecular imprinted polymers. Although the experiments that we're going to be doing as part of this GYAN series is going to be on an enzymatic sensor, but that's just to get you used to how you build a biosensing device. But if you were to start your research on biosensing and start by making biosensing devices, my recommendation, strong recommendation, would be to use molecular imprinted polymers. You can choose your own molecular imprinted polymers, right? But go for that one and then combine it if necessary, if you need a much more higher sensitivity or a lower limit of detection. Okay. Coming back now again to the biosensors, what are the different fabrication techniques? I mentioned a lot about screen printed electrodes, right? That's a scalable technology. I still don't understand if they are scalable technologies. Why are they still very expensive, right? Screen printed and inkjet, the people make out of screen printed and inkjet printing. There's also the PCB electrodes that's normally made. You also have micro molding. I gave you examples of micro molding yesterday, where I showed you how you can micro mold micro needles. I gave you the example where you can take a aluminum block and use electric discharge milling, make your pyramidal structures, and then pour in PDMS and peel it off, and then you get the reverse structures, and then you can bring in a photopolymer such as SU8, and then you can make the pyramidal structures there. Okay. You can also do electropolymerization, and you can also do nanomaterial functionalization. I think we've covered a little bit of all of this and as part of the fabrication techniques that are involved in building a biosensing device here. We haven't covered any optical biosensors at all, right? So I'm just going to give you a little bit of more information because we are covering the basic fundamentals for biosensing. So I think it will be nice to have this particular slide on optical biosensing. Optical biosensing could be, you can have different types of measurements. You can have one measurement where you're doing absorbance or transmittance measurements, right? This follows which law? Well done. Good for remembering Beer-Lambert's law, right? This follows Beer-Lambert's law, which is a simple law which says that absorbance is a product of concentration A equals ECL, right? I think I'm sure most of you have done it, or even if you've done the engineering, you still covered Beer-Lambert's law as well. All right. So absorbance, this is your Beer-Lambert's law. Here you have a source which is basically and then you scan. You then measure the absorbed or transmitted light here. You can get a response, which if it's an absorbance, you get a peak, and if it's transmittance, you normally see a dip. So you can normally see that in your FTIR spectrum, where you look at the transmittance versus wavelength there. So you have a dip there. And then you can do the analysis, and your absorbance is directly proportional to the concentration here. While with the transmittance, you get more like a exponential decay kind of thing there. You have transmittance and you have the concentration going down. So that's one method of doing an optical detection method. The other one is very common, which is fluorescence. Fluorescence is normally known to be very sensitive. That's why fluorescence is very common. And as I said, now if you use in this particular case, you have an excitation wavelength. You excite your molecule, and then you'll have an emission monochromator, and then you have your intensity measured against the wavelength of emission, and then you can get your plot of intensity versus concentration. Again, there's a good correlation between intensity and concentration there, following the same principles there. All right. Anybody remembers Jablonski diagrams that we had for fluorescence to explain fluorescence? No? Yes? Modified Jablonski diagrams, right? ISC and stuff like that. Yeah, all remember that? Interconversions and state like that? Good. Well done. Unfortunately, we are not going to be talking a lot about optical biosensors. We're going to this is just one slide to run about optical biosensors. Can anybody in this room tell me why is it hard, or why is electrochemical biosensing more useful when it comes to point of care and wearables? Okay. What's the biggest challenge? What is the number one desirable characteristic of an electrochemical biosensing device? Okay, fine. Forget about the specificity, forget about the performance. I gave you an electrochemical device. What is the most important characteristic that you need from it? Signaling, okay, yes.
Accuracy.
Accuracy, yes. Fine. Accuracy, specificity, sensitivity, that's all fine.
So cost efficient.
Cost, well done. Well done. Did this cost idea come from this Gyan series?
Comfortable to wear.
Comfortable to wear. Can you give me a synonym for that?
Tolerability.
Tolerable, flexible. The word I'm looking for, it starts with a P.
Portable.
Yes, portability. Right? So if you want something to be portable, you should be able to miniaturize the whole, right? So the problem with optical biosensors is it's not very easy to miniaturize to a dimension. Like for example, your electrochemical biosensors could be miniaturized to the extent that if you look at a CGM sensor, which I'll try to show you this afternoon, I will implant it in front of you. The CGM sensor, or in fact during the break I'll implant a CGM sensor and show you how the CGM sensor is implanted. A CGM sensor has got all the electronics at the back of it, right? Please don't ask me to look at my glucose readings because I've been eating very well here. So, but I'll definitely show you how the CGM sensors are implanted, right? So the problem is that you cannot miniaturize and have all the components in a very small real estate. You know, the term we use is real estate. You need a much more bigger real estate for the optical biosensors, and that makes it a bit more difficult to implement in terms of portability and on-site measurements and stuff like that. Although having said that, they're very sensitive. So you will find optical biosensors next time you go through the airport security checks. When they make you stand and do this and that and all different types of exercises, and they check your bag. They're basically using, they take a swab out of your bag and they look for things. Besides the X-ray thing, they also do the Raman measurements as well. And you would see that they take a swab and then they take it to a device there, and then they put the sample in there, and then they throw away that fabric, and then they use so they have a Raman system sitting in there. So it's nice in terms of performance, optical systems, but portability is a big issue there. I mean that's my understanding. Okay, transduction methods. Everybody happy with this? We covered all of this yesterday. I mean, I've got some few slides to show you exactly what it is, just to consolidate on amperometric measurements where you're measuring the current, potentiometric sensors where you're measuring the changes in potential, impedometric sensors where you're measuring changes in impedance, and voltammetric sensors again here. This is an example of slide to show you exactly how the signal response look like. So this is a typical voltammetry. You have cyclic voltammetry and square wave voltammetry. This is how a typical cyclic voltammetry signal looks like. As I said, you have this nice birdie-like structure. I mean, everybody who starts electrochemistry loves to see the first bird, a nice bird-like structure, because that gives a good indication about the health of your biosensing device. If you have your max and your oxidation and reduction peaks at the right place, your reference electrodes are working well, right? So this is how this is how a cyclic voltammogram looks like. This is a image showing you how a square wave voltammogram looks like, right? Again, you can see that these are all directly proportional, and that's how you normally get it for the analysis side of things. Amperometry, where you're measuring current, right? Measuring current over time. So this is here. This is your amperometry. This can vary. You can get steps. If the current varies, if the concentration varies, the current varies. So when we do continuous monitoring, you can have a profile which can last you over 24 days. I think that's what's normally done in a CGM device. And then you have got your electrochemical impedance here as well. As I mentioned, you have a sinusoidal AC potential signal here as well, which is good to probe your design architecture and everything like that, but it can also damage very thin films that you have. So you can then look into other methods such as differential pulse voltammetry and square wave voltammetry, which is given on your screen here. I won't go much into details. Everybody happy with it? Or does anybody want me to go over it one more time? Because I think we covered most of it. I'm just trying to consolidate it and just doing a quick recap of your electrochemical transduction methods. Everybody happy with it? Okay. All right, let's have a look at few of the electrochemical sensors, how these sensors actually look. And these are examples of some screen printed electrodes. If you had to look at how this would look like if it's placed in a beaker, this is what happens in a beaker. You have a working electrode, you have a reference electrode, and you have a counter electrode, right? And you're applying a current between the working electrode and the counter electrode, right? And you have a reference electrode which is there to keep the potential fixed, right? This is your reference electrode here. So how would you normally implement this? So talking about miniaturization, these are planar electrodes, right? These examples that you see here are planar electrodes, right? Again, you might be thinking, why is he teaching us so much about planar electrodes and biosensing technologies, although we want to learn more about wearables? All this technology that we develop here, right? When we are developing wearable devices, anybody who works on wearable devices will tell you they don't start directly with wearable devices. They start working and they optimize the protocols on planar substrates on materials that are much cheaper. And then this protocol is transferred to a wearable device. Therefore, it's very important that you understand the basic fundamentals, the instrumentation, everything that's involved in a point of care wearable, in a point of care biosensing device, so that you can then transfer it to a wearable device. And that's how I started my journey in wearable devices. I learned about biosensing devices first, working on planar electrodes and stuff like that, working on disc electrodes and stuff like that, and then I thought about, okay, how can I implement this now onto a wearable device? You will have differences there because suddenly your surface area will increase. Those are things that you need to take into factor, right? So gold may be suitable for as a planar electrode, but it may not be suitable as a wearable electrode. But those are minor tweaks. You can change the different materials. If you see that between gold and platinum, your platinum electrodes are performing better with your screen printed electrodes, with your planar electrodes, then you transfer that onto your wearable electrodes. All right. So that's something that I want you to bear in mind for those of you who are going to be venturing into this area of wearable technologies. The images that you see on your screen, these are basically 3 electrode setups. I mentioned you're going to have 3 electrodes which are most important. Where does the main action take place? Working electrode, counter electrode, and reference electrode, right? So this is how you have, and then you have an electrolyte bridge. This is basically a drop of solution that sits on top of this whole thing there. You can have, as you can see from the schematic here, you have your working electrode where the action takes place, your counter electrode which is a semicircle. And as I said, as a rule of thumb, people normally try to have a much higher working, either the counter electrode should have a higher surface area or surface area comparable to the working, similar to the working electrode. Mostly the we've seen the best case scenarios where you have much more higher. So if you look at platinum wires that are normally sold, they would have a lot of turns on them, a lot of knots. That's how they normally work. Okay. So that's the schematic, and this is then again how you bring that and how you implement this. So this is the first example of how we implemented our working electrodes, our planar electrodes straight to a microneedle array here. This is a schematic showing you that. So what we have here is basically you have the first layer, as I mentioned to you, we have SU8, which is out of which you make your microneedles. Yeah, and we call them microneedles or microspikes in those days. We were calling them microspikes, I think. And then on top of that you have a layer of gold. Right. So you have your SU8 and then you have your gold on top of it, and then on top of it you have your enzyme. Here, because you had gold, what we did here was we immobilized the enzymes using what sticks to gold? Thiols. Right. You remember that example I gave you of SH groups? So we had a thiol group there. So we had thiol, we form a self-assembled monomer layer of thiols, and then we do EDC and HS coupling. This is used to combine, this is used to get you amide linkages. So if you have a COOH group and you have a NH2 group, you can combine these two together with a condensation reaction where you end up getting, you lose a molecule of water and you get CONH linkages, and these are very strong covalent linkages there. So you have your self-assembled monolayer which are terminated with the carboxylic acid, and you have your enzyme which has got the amino groups. So your amino groups of the enzyme bind to the COH group of the thiolated compound, and then they are all self-assembled there nicely. And then on top of that you put a epoxy polyurethane membrane. This was the first studies that were done, but these were done using again, these were done in vitro, right? So what we had here is a microfluidic system. This was a paper that was published, and you can see from here that you have different concentrations. You keep on adding it because it's a microfluidic system, so you're doing continuous measurement. This is an example of chronoamperometry where you're measuring current over time. So what you start here, your currents drop down, both of them. There are two channels here, right? There are two different devices here. So you add then 2 millimolar, you get a step, then you add 5 millimolar, you get another step, then you add 10 millimolar, you get another step, 15 millimolar, 20 millimolar, 25 millimolars. So you can see there are two different devices. The reason you have different performance is because you're using a microfluidic system and you're pumping fluids at different flow rates, right? So you can see that this is such a much higher flow rate, and you can see here that these are the different steps you're getting here. All right. So this paper has been published, and this is also one of your suggested reading. This is just to show you what I just mentioned before, that once things work on the planar electrode, you can then try to transfer them onto to your wearable device. I call them microneedle here. Microneedle basically is a glorified subdermal wearable sensor, right? So you can easily transfer them, and you may have to do some tweaks, but it's something that can be transferred, and not only you can also publish them. And then finally, the other thing that we need to look, I think we are running a little bit of time over time, but I will be talking about performance metrics here. So I've already given you some examples yesterday. Do you remember the Clark grid errors, anybody? What did we use for the lactate sensors to look at the performance? Louder please. Yes, somebody said BA. What does that BA stand for? Bland Altman plots, right? Okay, good. So I'll just quickly go over this thing. So how do we look at the performance metrics? How do you look at the performance of your sensors here? Well, the important criteria or the important things that you normally need to report in your paper are sensitivity, limit of detection, selectivity, stroke specificity, response time and stability, calibration and reproducibility. These are the important metrics that you need to know and understand if you're going to be working on your biosensing devices, right? What are these? Sensitivity. I've shown you many of those calibration curves. You remember dose response curves, calibration curves? What you do there is you have different concentrations. You measure the response or the current in response to these different concentrations, and then you do a plot. You do a fit here, right? And you see how far this particular slope is your slope of high sensitivity, right? And this is your here. This half of this is what you call as the limit of detection. When you have a low sensitivity, that's your limit of detection. This is where your blank signal is. This is where your blank signal starts. Your blank signal is never zero. Your blank signal will have a background current there. This is where the blank signal starts. So this is the dynamic range. This is your linear range. This whole thing, how far you can go, how far you can see the increase, is what your dynamic range is. And after this point, it starts to saturate. You can see it starts to plateau there. Right? So these are how you calculate your sensitivity and your limit of detection. The slides, you will be getting a copy of these slides, so don't worry if you're not able to get these slides, but this is something that you need to characterize for your device. I'm not here and I'm not expecting all of you to absorb everything, because I know I speak really fast and it's virtually inhuman for me to expect you to absorb everything. But please do have a look at the slides after you finish, when you have some time in the evening or whenever you look at the lectures, because this is important piece of information that you need to know about your devices, the performance metrics of your devices. What is the lower limit of quantification? What is the higher limit of quantification? What is the limit of detection? What is the sensitivity? What's the linear range? What's the dynamic range? At what concentrations do they plateau? When does it start to saturate? You need to know all of these about your sensors before you can bring them into the real world, before you can do any translational research. You need to optimize, and even with in vitro studies, those of you who work on in vitro, stop working on PBS. Identify what your final sample matrix is going to be. Are you developing wearable sensors? Okay, so you're going to be working on skin interstitial fluid or sweat. Why don't you use artificial sweat? Why don't you use artificial ISF? You have protocols for all of these available in the literature. If you can buy blood as a reagent, if you've done the training and everything, if you got the research ethics approvals, use blood, use whole blood, use plasma, use serum, and then develop it, because what happens is if you develop everything in PBS, phosphate base, in 7.4 pH and things like that, you're basically once you start working in real samples, you will have to go back to the drawing board and redesign your sensors, right? So very, very important to do all your studies right from the start in something that is very similar to the final application area that you're going to be working in. Okay. Talking about selectivity and specificity, good examples here. Selectivity is more when you have single analytes, how specifically it binds to it. Selectivity is how selective it is, how good selectivity is. Normally when you have sensors, for variable sensors, you want something which is it shows you good selectivity but not very good specificity. Why? Because you wanted it to bind and then you want it to detach, so that you have those sites being regenerated, right? So this is a slide, and there's a paper here which explains selectivity and specificity very nicely, which can also be seen from this particular diagram here. I've given you the example of antibodies here, and these are more like molecular imprinted polymers, right, to give you examples of sensitivity and selectivity that you have on your screen. And then response time, how long does it take to respond to the change in concentration? That's a very important parameter. You don't want something where the response time is, it takes about 15 minutes to get the actual value, right? Okay, the incubation step may take 30 minutes, but once the incubation is done, how long does it take you to get the sensor? Because think about it, you're developing devices. If your devices are going to take 4 hours, 5 hours, then it kills the whole purpose of using devices at point of care sites, right? So you need to have devices which have got a very short response time. Then the other thing that you need is stability, performance of the device. How does the device perform? Has it got regular, you measure it over a certain number of days and you see how the performance is over days. Is it a stable performance or is it a unstable performance? One day it's giving you high value, one day it's giving you low value. You normally measure the baseline. So this one is an example of unstable one, and this one is an example of how the values are very stable. There might be slight variations, there might be slight degradation, which you can account for. If there is a trend, you can bring in the machine learning and the AI tools, but if there is no trend there, nobody can save you there. Does that make sense? Okay, and then finally looking at the calibration. I mean, we've been through many of these. I've shown you I don't know how many of these calibration curves. This is how you get your calibration curves. Look at this nice calibration curve that you have here. Look at these ones here, right? What are these calibrations? So the one thing is calibration, right? This is where which is more like a dose response curve, right? The other one is here. This is an example of, this is the kind of data you would expect in a journal, right? To get something without error bars, if you cannot publish, if you don't have error bars in your data, you need to have n equals 3, and you need to have multiple points, and you need to have error bars. If you don't have error bars in your data, you cannot publish your data. Simple as that, right? Any journal, any paper that comes to me for review which doesn't have an error bar, I know that is made up data, because in real world you always get some variation. Even if it's a minor variation, you get some variation there. So this is the example of error bars shown nicely in the data here. And then talking about reproducibility, I mean, look at this. Day 1, measurements done over day 1, day 3, day 7 and day 14. Right? This is clearly an example of a continuous glucose monitoring sensor here. I mean, there's slight variation there, but by and large, look, it's very, 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 very reproducible. Right? That's nicely shown in this particular data. And this again is nicely described in this particular paper for which I've given the DOI in the slide here. All right. I think that brings us to probably the last or second last slide, which is the Clark grid error. You remember this Clark grid error? Everybody remembers CGE? Clark error grid or Clark grid error, right? This is basically, as I said, when you're looking at your measured blood glucose with your biosensor and looking against a reference method, which is a gold standard, if ideally all the values should fall on this straight line. The nice thing with glucose sensing is there's a little bit of a room to maneuver, right? And this room for maneuvering actually gets narrower as we move from higher concentrations to lower concentrations, because as you move towards lower concentration, there is less room there's less margin for error, right? And as you can see, because we do not want a case of hypoglycemia, we don't want our sensors to be overreading when the readings are down, right? And sending this. So this is the Clark grid error, which is mainly used for glucose sensors. And then finally Bland-Altman plots, right? We covered this as well yesterday. Ideally all the points should fall on this straight line here. This is again looking at the difference between lactate and the average lactate here. Ideally everything should fall along the straight line here, and these are your confidence limits. You have 95% confidence limits. As long as the points of majority of the points are falling between these two blue lines, everything is fine. These ones are the outliers which need to be I mean in this case there's only 2 outliers or maybe 3 outliers which need to be investigated, but you can call them outliers and just leave them alone. But if you have many points which are outside beyond if you have more points outside the blue lines, then that's a worry, then your sensors are not the best, right? So you need to think about that as well. And these Bland-Altman plots are normally used for looking at other types of sensors other than glucose, lactate, ketone sensors, and all the other things are normally done using Bland-Altman plots. You also have something called as violin plots that I'll show you when I show you the p-tau results. So kind of now almost towards the end of the talk here. So when you move from the lab to the clinic, there are certain factors that you need to account for. Biofouling is a big problem. And as I said, if you understand the trend of biofouling, you can bring in your machine learning and AI tools and you can use that. I showed you yesterday a case study where we used AI tools and we could make much better sense of the data. Miniaturization, how do you miniaturize? I mean I showed you I haven't done that, but I have one lecture just dedicated for animal studies to show you the challenges there, because nobody has done animal studies, nobody has used wearable sensors on animals, right? And the animals actually deserve more to have wearable devices because they cannot give you a visual assessment feedback and you cannot have capillary blood from them. So there's a lot there that needs to be done in terms of miniaturization, and then as I mentioned you need to have the right approvals and how do you integrate them into these apps. I mean this is the latest fashion these days. So this is my last slide. What we've done today is we've recapped on biosensor types. I hope I don't have to repeat it again. I hope I've helped you consolidate all your learning that you've been doing over the last 4 lectures. We've seen the values, the importance of translational value and the challenges that you have when you translate a device from the lab to the clinic. And if you have any questions, I'm more than happy to take them. I'm sorry for running over time.
It's okay. Any questions?
Actually, sir, in the molecular imprinted polymers,
Yeah.
you have mentioned we don't require washing steps.
No.
So, sir, how we can regenerate the sites there to bind another antigen? How we can reuse those devices?
Excellent question. These are point of care devices. When I say point of care devices, we are looking at disposable devices, number one. The other thing you need to understand, clinical samples, you never reuse the devices.
Okay.
I would never reuse a PCB electrode, no matter however good cleaning protocol I have, if I'm using it with human samples, because there's every chance of having cross contamination. So you don't need to regenerate it. You can just dispose it off.
Okay. Thank you.
Yes, but this question, the same question with wearables would be very, very interesting. So in that case, you can have wearable devices because at certain point they will get saturated. Then you can change the device and use a new device, or choose molecular imprinted polymers that can bind and unbind. So as I mentioned, between selectivity and specificity, that are specific but less that are selective, so they go and bind, but then they unbind later on, so that you can free the site, and then you can have more. You can do more continuous measurements. But ideally, you would like to have something that works over 6 hours or 12 hours, and then you dispose them off.
So for removing antigens, we require again the some kind of proteins and enzymes, right?
For removing what?
The analytes which got binded into the sites. So to remove them, we require enzymes and proteins.
No, we don't need it.
So how can we remove them?
No, for wearables, you've used it. It will give you a value, and then you dispose off and get a new one, a fresh one.
Okay.
Yeah. Good. These are really good questions. It shows that you're thinking about it. Any more questions? No? Everybody's eager for a cup of coffee or tea?
Yes, any more questions? So I might have a few. Actually, so this molecular imprinted polymers, they seem like a very good method of sensing. So is there any limit to it? Like you have tested it with vancomycin and any
We've
other-
tested it with
so
vancomycin.
and some protein.
Yes.
Yeah. So what I mean, can it detect any analyte in the world or
In
target?
principle, yes. With
In
the right
principle.
choice of molecular imprinted polymer.
Okay.
In principle, yes. Because
Okay.
there's a wide range of polymers that are available.
Okay.
It depends on your target analyte, how
Size of the
hydrophilic,
target.
hydrophobic it is.
Okay.
So you have to be very careful. That's why I stressed a lot on the interaction mechanism. And I try to explain it by you explaining the different forces that act, the hydrophobic forces, the electrostatic forces of attraction, the hydrogen bonding. So you have to actually, it will be nice to have this whole thing modeled nicely,
Yes, yeah.
to have a model which can help you then choose your polymer accordingly.
Okay. Okay. So first you model it and then check out.
Yeah, you
Then
model
you-
it and you model it with, so you will have polymers which are, this polymer is suitable for hydrophobic molecules, this polymer is suitable for hydrophilic molecules. The thing is with electrochemistry, you have to have it it has to be very aqueous based.
Yes. Yeah.
That's where the challenge is. That's where you bring in zwitterions and all those things.
So what, like for the benefit of the participants, what sort of tools, modeling tools do you generally use for this? Just few, if you can name
you
a few.
can just use the Gen AI tools that are available. You can use anything as simple as ChatGPT and DeepSeek and Gemini if you want to just understand it first of all, and then you can use PyMol.
Okay.
And the reason I say that, it will save you a lot of time to model it, to understand it through the generative
Traditional,
AI
yeah.
tools, and then it will suggest you how to model it using the PyMol and stuff like that. That saves you a lot of time, because I know that this can be very time consuming when you sit down there and try to model and then you try to identify the sites. It's very hard.
Yeah.
Why not use the tools that are available at your hand, at your disposal?
Okay. So these violin plots you will be showing later on, I guess.
I'll be showing you tomorrow.
Okay. So in this lecture, just like as sir has said, we covered different biosensor types, especially the bioreceptor sensing using MIPs, molecular implanted polymers. And then we also looked at very important aspect is if you develop a new biosensor, then you have to look at all the plots, Clark grid error, blind Altman, and all the characterization. These are the typical characterization steps, calibration that you need to follow for your own biosensor to show and to understand that whether it is working successfully or not. So with that, we end this session. There is tea outside, so let's reconvene again after a short pre-break, approximately around 11:45 or 11:30. Thank you.